All right, right now we're going to take the national anthem and we're going to do this in the standing ovation. This will be conducted by the public affairs.
Nigeria, we hail thee, our own dear native land. Those ranks tongue may differ, in brotherhood we stand. Nigerians are proud to our sovereign motherland. Our flag shall be a symbol that truth and justice reign. In peace, all bow to honor, and this we can escape. To hang on to our children, our banner without stain. As long as we proclaim, from hills and valleys we've come, to draw from the stream of knowledge. Our duty is to succeed in learning and service. Excellence is our goal. Ahmadu, Bello, University. Yeah, our dreams we shall achieve. We give thanks to Almighty Allah. In Him we derive our strength. We beautifully shall abide by the ways of good learning. North to south, east to west, your nature is wisdom. Ahmadu, Bello, University, our citadel of great learning.
All right, thank you very much. You can have your seat. Okay, right now we're going to have an opening remark by the Dean of Faculty of Life Science. Okay, thank you for that. We're going to have a welcoming remark by the head of department of Botany, that is in person of Professor Ahmadu E. Ali. Yes, ma'am.
The Vice Chancellor, sir, ably represented by Professor Raymond Bako, the heads of departments here present, the Dean of the Faculty of Life Science, the Deputy Dean of the Faculty of Life Science, Assistant Dean, distinguished guests, esteemed colleagues, ladies and gentlemen. It is with great honor that I welcome you all to this groundbreaking workshop on plant identification, herbarium digitalization, and AI modeling. Today marks a significant milestone, not just for the Department of Botany, but also for the broader scientific community that values plants as the foundation of life, biodiversity, and sustainability. The Department of Botany, Ahmadu Bello University, has a proud history of advancing plant science through teaching, research, and community engagement. Yet, we recognize that science is dynamic, and to remain relevant, we must embrace innovation. The workshop embodies the spirit of transformation, bringing together classical plant identification with modern digital technologies and artificial intelligence. Plant identification remains the bedrock of botany, ensuring that we understand, classify and conserve the rich diversity of flora. Herbarium digitalization allows us to preserve centuries of botanical knowledge while making it accessible to researchers worldwide, thereby breaking barriers of distance and time. While AI modeling represents the frontier of plant science, enabling us to predict, analyze, and innovate in ways that were unimaginable just a few decades ago. By integrating these three pillars, we are not only safeguarding our heritage, but also preparing for the future, where plant science will play a critical role in addressing challenges such as food security, climate change, and environmental sustainability. I commend the team of organizers, the facilitators, and participants for their dedication to this vision. Your presence here demonstrates a shared commitment to advancing knowledge and ensuring that our department continues to lead and innovative research. As we embark on this workshop, I encourage all participants to engage actively, share ideas generously, and build collaborations that will extend beyond the workshop. Together, we can transform the way we study and apply plant science for the benefit of humanity. On behalf of the Department of Botany, Ahmadu Bello University, Zaria, I warmly welcome you to this plant digitalization workshop. Thank you.
Come on, let's appreciate you very well. Let's appreciate it. Thank you very much, Ma, for that opening remark. That's a welcome remark. Now I will call on Professor Mariam, that is the Dean of Faculty of Life Science. Please, let's jump to sense. Let's jump to sense as she gives us the opening remark. Thank you.
Good morning. The representative of the Vice Chancellor, Professor Raymond B. Bako, the Deputy Dean, the Assistant Dean, the Head of Department of Botany, Professor A.K. Ademu, Professor Dangora, all distinguished guests, esteemed resource persons, respected colleagues, and dear participants. Good morning. It is my pleasure and honor to welcome you to this plant identification, herbarium, digitalization, and AI modeling workshop hosted by the Department of Botany in the Faculty of Life Sciences. This workshop would not have come at a more critical time as global biodiversity faces unprecedented pressure. The role of plant scientists, taxonomists, and conservation experts is more essential than ever. The work we do in research, teaching, and field science must evolve, and today's event reflects that evolution. Combining centuries-old herbarium practices with emerging technologies artificial intelligence is very important. Over the next 7 days, you will not only learn how to identify and digitize plants, specimens, but also why these skills are vital to conservation, policy development, and future scientific issues. I commend the organizers of this event for assembling these experts whose combined experience span taxonomy, AI application, field botany, and conservation science. The inclusion of field excursions and hands-on visualization exercise demonstrates that this is not just another academic gathering. It is a practical, forward-looking training that is designed to to empower every participant. I urge you to take full advantage of this rare opportunity, ask questions, build networks, challenge existing methods, and imagine new possibilities. Our region, and indeed our continent, needs more scientists who can bridge traditional botanical knowledge with modern digital tools. You are the next generation of such scientists. To our facilitators, thank you for your commitment to capacity building, to every participant, whether student, curator, researcher, or industry professional. Your presence here is an investment not just in your career, but in the future of plant biodiversity in Nigeria and beyond. So I say thank you. Let this week mark the beginning of new collaboration for us, new research directions, and new leadership in plant science. On behalf of the faculty, I want to declare this workshop open and wish you a productive, inspiring, and enriching experience. Thank you.
Thank you very much, Ma. Please, let's appreciate them more. Let's appreciate them. All right, I'm going to call on the representative of the Vice Chancellor in person of Professor Raymond Bako to come and give us an address and official opening of the workshop. Please, let's jam our hands for him. Thank you very much. The Dean has officially opened the workshop, so she has made my work easier. So thank you very much. The Dean, Faculty of Life Sciences, the Head of Department, Department of Botany, the guest lecturer, Professor Michael Delawa. Nice to see you again. Our very senior colleague, Professor Ademu Kasim, professors here present, academic colleagues, students, distinguished ladies and gentlemen. I welcome you to this very important workshop on plant identification, herbarium digitization, and the application of AI in plant characterization. I bring you warm greetings from the Vice Chancellor, Professor Ahmadu, who will have been here personally, but he asked that I represent him at this meeting. Let me say that the Vice Chancellor and indeed the present University Administration places a lot of emphasis on academic discourse and capacity building. And so the Vice Chancellor and the administration is always very excited to see that units of the university, faculties and departments are making efforts to organize workshops of this nature, to share knowledge and to build capacity. And so the university administration will continue to support these efforts. I will continue to encourage departments and faculties to organize workshops like this. You can be rest assured that, as has been said, that this workshop provides an opportunity and a platform to engage intellectually, to network, and to share experiences. Ahmadu Bello University is known for intellectual rigor. So we hope that this platform will provide that opportunity to deepen existing knowledge, to question established norms and established bodies of knowledge, because that's the essence of the intellectual tradition. So on behalf of the Vice Chancellor, Professor Adamu Ahmed, I join the Dean, Professor Ahmadu, to declare this workshop open, and I hope that at the end of the day, we'll leave here better educated, better equipped to carry out our work in the area of botany. Thank you. God bless us.
All right, thank you, sir. Please let's appreciate him. Let's appreciate him. Thank you, sir. All right, next on the item is launching of plant press. While going through this program, I can see that it's a man of capacity that have been made to launch this plant press. The former Vice Chancellor of IBB University, the person of Professor Ike Adamu. Please, let's jam our hands together as we welcome him on stage. Yes, sir, you're welcome. You're welcome, sir.
The Vice Chancellor, Ahmadu Bello University, ably represented by the EVC academics, Professor Raymond Bako, the Dean, Faculty of Life Sciences, Professor Mariam, the Deputy Dean, representative of the Dean Physical Sciences, the Head of Department, guest speakers, invited guests, participants, ladies and gentlemen, good morning. Let me once again appreciate the department for this noble objective of training and the training of scientists. And I'm sure most of us in the last 20, 30 years, when you are coming to Zaria, there are certain species of plants that serve as markers, Siba pentendra, that once you arrive and see, even in the night, you will say, yes, you have arrived Zaria. But these days, they have all been wiped out, they've been caught, and there are no replacements. It is through these particular workshops that such sustainable SDG goals can be revitalized, because you need to identify suitable plants that can survive in an ecosystem across board. Coincidentally, in the country now, especially in the north, the issue of tree planting has completely died down. And we are making a very serious mistake, because the desert encroachment is fast approaching devastation of the climate change. So what we are doing today is part of the SDG goal, the sustainable development and the environment. And I'm sure we have able guest speakers, we have the participants that can take us through and see, through this particular workshop. Coincidentally, when I came in, I was discussing with the VC. When I said, is it a launching or unveiling? He decided to correct me to say it is unveiling, not a launching. Otherwise, probably may put in at the third corner. I don't know that he seek approval of the VC for him to launch. So with the kind permission, the VC is going to be unveiling, not launching. Maybe later I'll meet you in the office and discuss for your support personally. So do we have the plan present? How many do you have? Well, this is an innovation of the staff of the Department of Botany. For the typical plant press, we know what it is. Usually a very heavy, what I call it, steel, not comfortable, but the department staff, Suleiman Etam, HOD, and others were able to innovate this and come with a portable plant press. Unlike before, you can see it's something that you can go to the field, collect your materials, package them back. You can collect as many as possible. You can collect as much as possible and then carry them like your handbag. So you are a field worker, you can carry them like a field rather than looking for something to take on your head. Or you can even carry them and walk gently that yes, I'm a scientist. I've collected plant species from the field. So let me congratulate the staff, Suleiman et al. For this innovation. This is what we need. Is he around? Dr. Suleiman? Okay. So maybe all of us at the high table, we come down or we know where we are. Or the umbrella and then yes, please give each one one.
Not so that is switching camera. Yes, a little bit. Come down.
So on behalf of the Department of Botany, they, the 17th of November 2025, were unveiling this innovative plant press for collections of specimens in the field. The glory of the Almighty God and the benefit of the humanity. Thank God, God bless. You have to show.
Calm down. Hold it together.
You hold together. You are holding it. So come on, hold on. Let her hold it.
Go. Okay. Yes, I'm ready. Go. I think you should have ended. Okay. Thank you very much.
I think the dean is suggesting rather than going back, we should take the group photographs. The representative of the VC is ably here and wants to continue with some other assignment. But my friend coming. I think just coming over.
That's okay.
Okay, ready? I think again, 1, 2, 50. Thank you.
All right, thank you very much for that group photograph. The next thing on the list is keynote presentation. And our speaker here is Professor Michael A. Adelawa, who is going to be speaking on modern approaches in plant identification and plant biodiversity studies. Ladies and gentlemen, please join your hands together as we welcome Professor M.A. Adelawa. Okay, I know some of us know Professor Adelawa, why some don't know. He used to be staff in this university before he transferred his service to Federal University, Lokoja. So I know he's going to do most of the introduction. So please let's pay attention to him. Thank you, sir.
Good morning, everybody. I want to use this medium first to appreciate the head of department for counting me worthy to take this keynote address and be part of this great occasion. I don't take that for granted. Thank you for making me a part of this program. I don't think I need to introduce myself again. We have been together, so it's like coming home again to meet my people. I believe this time we will have a very great time with each other. So thank God for everybody. At least I can see everybody bubbling by the grace of God. I pray that God Almighty will continue to keep us as we relate with one another by His grace. Sorry, I'm still trying to open it. Okay, thank you so much. Good morning, everybody, once again. Professor Adamu, good morning, sir. My dean, good morning. Yes, sir. So, I appreciate you all. I'll be speaking on a plant identification and preservation technique. And we should know what, before you even identify, you have to make a collection. And in those days, I used to tell my students that when you want to collect, first and foremost, you must know what to collect, what you want to collect. And you must know where to collect it from, and even how to do the collection. So, as I have said, we are looking at modern approaches in the area of the plant identification and the preservation technique. As we know, plants are the foundation of life, even on Earth, as they give us oxygen, food, and medicine, also shelter, and countless of other resources that sustain our existence. But despite their importance, our plant resources face increasing threats from deforestation. At least we know that from the report we get on this Amazon forest, which happen to be the largest forest reserve we have in the whole world. We know the state now is below 10% now. The result of human activity, which is deforestation. We also have urbanization and climate change and neglect from the government and people. So to protect what remains and to restore what we have lost, we must begin with the knowledge of what plants exist, where they grow, and how we can preserve them. And like I said earlier on, this begins with the science of plant identification. And also, what I used to tell my students in those days, that when you want to identify a plant, after picking a plant you don't know from the wild, how do you go about it to identify it? For example, I give an instance. You are coming to this place and you saw an ID card on the ground, and you pick that, and you know that ID card belongs to somebody within this auditorium. You have to enter with the ID card and start looking at faces one after the other. With the ID card in your hand, you look at the first face. Oh, doesn't resemble this one. Look at the second one. And what you are trying to do is to identify that particular person in that ID card. Immediately you get to the person that owns the ID card, within the population we have there, you have already identified that person. You may not know the name of that person, but at least you fulfill that aspect of identification. So I used to tell my students, after picking an unknown plant from the wild, you want to identify it, there is a place where we have the repository of plants, which is the herbarium. You have to visit the herbarium and start searching one after the other, looking at the folders that we have in the herbarium. And until you start looking at it, we call it identification by matching, one after the other, until you get to that plant. Though it's a tedious process, and that is why we are looking for a modern way of doing it easily. So like I said earlier on, before any form of identification, the first thing to do is to collect the plant specimen first. And we know all the equipment we use in plant collection, like secateur, long hand sickle, a knife. Those in the herbarium, we know what I'm talking about when you want to collect. Also what to collect, where to collect, time to collect. Plant samples are very, very important for identification. So if you don't know what to collect, then definitely you may have problem. You collect from the population or a single individual. And we used to discourage this. While you are collecting, don't collect where you see just a single plant growing. You may not know that that may be the only one single plant that is remaining in that locality, and which is against conservation. If you pick that one and you remove it, then that is the end of that plant in that area. So we normally encourage people to pick from a population of growing species. When you pick from there, you are sure that whenever you come again, you will still meet those plants there. And also, where to collect, where do you collect from? And also time of collection are very, very important. Time. You don't go in the afternoon and make collection in the afternoon when the sun is very high. What will happen to the plant? It can quickly dry up, and the purpose of identification is defeated. But normally we can collect in the morning and also do identification after collection. And apart from collecting in the morning, we collect also late in the afternoon from 5 upward when the scorch of the sun has gone down. That time you can still collect. And we keep them in a polythene bag or leather bag to prevent dryness before we do our identification. The purpose of this is at least when you want to identify, you still have the whole plant standing. Because if you allow it to dry, it will be very, very difficult for the person who wants to identify. Like what to collect. Some students are fond of going to the field and collecting just a single leaf. When you collect a single leaf and you take it to the herbarium for identification, then it becomes a problem. And we normally advise that while collecting, you have to collect either with the floral parts or let plant that has a fruit or flower, with fruit or flower. Any plant without the fruit or flower, we call it what? A sterile collection. Like we used to tell our students, is a sterile collection. And if you go to the herbarium with a collection without a fruit or the flower, it will tell you, sorry, we cannot identify this. Go and collect it. So definitely we cut part of the branches that contain a floral part for identification. And like I said, time is very, very important. And you can identify even at the point of collection or later after mounting, even in the herbarium press. Then, like I said, identification can be done after fresh collection or after drying and mounting of a plant specimen. So plant identification is more than just recognizing a tree or flower by its local name. It is the systematic classification and documentation of plant species based on their morphology, anatomy, and genetic characteristics. Through proper identification, we can distinguish between species that may appear similar but have ecological roles. I'll still come back to that one, especially those species are similar. How do we identify them? We can discover new species that can contribute to medicine, agriculture, and industry. We can also track invasive or endangered species to ensure balance ecosystem. In the academics and practical fields of botany, agriculture, and environmental science, accurate identification forms the foundation for research, conservation and sustainable resource management. So if you want to do any research on a particular aspect, then definitely you need to identify the plant you want to work with. I remember the time I was running my PhD program at the University of Jos. This day I sat down in my supervisor's office. I was working. Then Professor Akweshi brought in a plant. He said, look, see this plant. If you can identify it, I will give you your PhD. I look at it, I said, this is a challenge for me. I said, Prof, sir, maybe before you come back, I will have gotten you. So he left. Then this comes to the advice I used to give to my students. For real identification of a plant, it's better you do identification by practice. As you practice this daily, using Nigerian flora, then definitely you know many of these plants. Most of the plants I know today, that was how I happened to know them. By practicing it, if you have Nigerian trees with a colored illustration, you can pick and come and check. After checking from time to time, so by now, for a student doing this, by the grace of God, within the shortest time, he will be able to identify maybe 50 to 100 different plants, different species of plants, both trees, shrubs, and herbs. So it's daily practice. It's daily practice. What you do daily, before you know, you become a master in it. So that when you get to the forest, or you get into an area, all the species that are there, and as we are going every day, at times I'll be working with my children. As we are walking, I'll be telling them, this is as a director in the car. They'll be looking at me, Daddy, what are you saying? When we move again, I'll say, this is Albizia Lebek. They will say, Daddy, which one again is Albizia Lebek? I'll start telling them, that's how these things started staying there. Till today, those things are still there, because it has become part and parcel of me. I practice it daily, and that is what I encourage my students to do. And people who are interested in this identification or naming of a plant. So like I said, accurate identification form the foundation for research. So by the time Professor Akweshi left, before coming back, I look at it and say, this one belongs to the family Liliaceae. Yes, I can assure that this is Liliaceae. Or the name, I started thinking, is Crinum. That's the genus Crinum. And I look at it, which species? I say, this is Jagus. This is Crinum Jagus. So by the time he came back, he was surprised. He said, sir, this is Crinum Jagus. He went and searched it. He came back and said, it's true. After searching, the picture came out and he looked and said, it's true. You are telling me this is Crinum. And he said, I will give you your PhD. I laughed. Praise the Lord. Sorry, I know he was not my supervisor then. So what are the techniques for plant identification? Plant identification techniques have evolved significantly from traditional methods to modern technological approaches. Okay? So we start with the morphological techniques. These involve observing physical features such as leaf arrangement, vernation pattern, flower structure, fruit type, and also bark texture. Herbarium specimen and field guide remain invaluable for this purpose. So we need to know the physical features such as the leaf arrangement. What is the leaf arrangement? Is it a simple, or is it opposite or alternate? We need to know the vernation pattern, whether it is reticulate or not, or parallel. We need to know the flower structure and the fruit type and bark texture. So we also use what we call the taxonomy keys. Taxonomy keys, actually the simplest one of the taxonomy keys. Though we have dichotomous, which is also an artificial system. We have dichotomous key, we have the number keys. But the one that we normally use most is this, that I believe is the simplest, is the number keys. So if you know characters that we are talking about, you know when a leaf is simple, you know when a leaf is compound, you know when a leaf is ovate, you know when a leaf is obovate, you know when venation is parallel or reticulate, you know the flower structure, then definitely you can use this key. Dichotomous key example, like we have the bracketed keys and also the indented keys. Those ones is just to accept, and also there are leads there for us to guide us. A plant is annual. You will know whether that plant is annual or biannual, or whether a plant is annual or perennial. This will guide us. But when you get to the number keys, which I believe is the easiest, and you know all these characters we talk about, is only to state, if the character is there, you pick one, write 1. If that character is not, you put 0. That is for the first character. You go to the next character, maybe of the leaves. If that character is there, you put 1. If it's not there, it's 0. So from this 100, 100 number, they are coded. Then you go to the number key. So each of these coded numbers leads to particular species of plant. We have those that lead to genus. So we have number keys to genus, we have number key that leads you to species. So number key happen to be the easiest one to use out of the taxonomy keys. We also have microscopic and anatomical studies, like internal structure like stomata, the trichomes, vascular bundles can also provide clues to plant identity. And another one is a cytotaxonomic approach, which is also an aspect that I love so much. Like certain chromosomes are peculiar to certain groups of plants. So if you see, for example, a plant is having chromosome of a 2n, a diploid number of 2n equals 16, you know that it belongs to family Fabaceae, and you can still trace it down to Protularia species. They have 2n equals 16. And also we have 2n equals 13. Sorry, 2n equals 26, n equals 13. That one belongs to the Acacia family. So once you know chromosome number, then you can. And this approach was the one I used one time during my study. I discovered the groups of lilies. There are 3. We have I'm trying to collect their name now. What's the name of this? The Dipcadi species. Dipcadi is the genus of those group of lilies. We have the Dipcadi longifolium, we have the Dipcadi filamentosa, and we have the Dipcadi tacasianum. The 3 of them look alike. They are like triplets in the field. For just an orthodox taxonomist, a confusion is there to pick this for this, to pick this for this. And how do you resolve that this one is longifolium, this one is tacasianum, this one is filamentosa? They look alike. The leaves structure are the same. The flowers are the same. The seeds are papery black. If you look at the bulb, 3 of them are bulbs, just like onion. So then how do we differentiate them? Then I have to use a cytotaxonomic approach. Going into the chromosome number, I discovered that longifolium had 2n equals 24. 2n equals 24. Tacasianum had 2n equals 12. And filamentosa had 2n equals 12. So I've been able to split them into 2. These other 2 groups are having 2n equals 12, while one is having 2n equals 24. I push that aside. This one is the Dipcadi longifolium, based on the number of a chromosome. Then the other 2, how do I resolve this problem? I still went in further. to do what we call karyotyping. Split the longest chromosome and the shorter chromosome. Look at the centromeric position. They are still the same. We have 3 longer chromosomes and 3 shorter chromosomes for the 2. Then I look at it. Okay, let me look at the last alternative, is to measure the chromatin length. And this I did using the eyepiece graticle. After measuring them, I discovered that T. cardi filamentosa had 89 μ. It's 89 μ in length, while Tacasianum have 79 μ in length. So with cytotaxonomy approach, we can split them into 3. That is, identify them as 3 different species. But for a layman, you will feel that they are all the same in the field. And that is why we use this approach to split plants into different species. We also have a cytotaxonomic approach, which is when we know it's a known fact that within the same family, some species of plant have the same chemical components. And that is the basis for research. Once you pick this plant and you're able to get maybe tapioca or any other saponins in this plant, then the other plants within that group or species within that genus, we also have the same chemical. They say blood runs within the family. Okay, so we can also use chemotaxonomic approach. Also, coming to the newest aspect, which is we have the molecular and the DNA barcoding. That is, advances in genetics have enabled scientists to identify plants through their DNA sequences with a high accuracy. So through DNA sequence, plants can also be identified, belong to species or different genus. And lastly, we have the digital tools and the AI application, which is the newest. So mobile application and database like a PlantNet, iNaturalist, and GBIF are empowering both scientists and citizens to identify and record a plant species globally. In fact, I am also using that, but it also has its own problem. Like I was discussing with one of our staff before this program started, that there is this plant we collected, they call it sugar plant. So when I tried to get the name, the app gave me stevia, rebaudiana. Okay, stevia, okay. We resnap it again, send it again. He gave us another name. What happened? We resnap it the 3rd time again. He gave us another name. So we have 3 names being given for the same plant. In fact, I'm still struggling with that now. I don't know what to do. But we stick to the first one, which is stevia rebaudiana. That was the one we still pick using now. Okay? So that is that with plant identification. So I won't go deeply into how we use all those. Maybe another person will take that aspect of AI and the other app being used. But secondly, I want to go into the aspect of preservation and protecting our green heritage. So once a plant is identified, the next crucial step is preservation. You need to preserve that plant. During those days, we say we preserve so that posterity will not judge us. What you have now, you can still get there and still use it in future. That is ensuring that plant species, especially rare and endangered ones, are also protected for future generations. So there are 2 main strategies that we use in preservation of plants. The first one is in situ conservation. That is protecting plants in their natural habitat through establishment of national parks, botanical sanctuary, and forest reserves. This allows species to continue to evolve naturally, and that is why all these areas need to be protected. If not, people will destroy those parks, they destroy those botanical sanctuaries, and even forest reserve to stop people from destroying them because of all the bioresources we have there, so that the species will continue to evolve naturally. This is the first one, in situ. Then the second one is an ex situ conservation approach. And these involve preserving plant materials outside their natural environment. Through seed banks, tissue culture, botanical gardens, and herbarium collections. And we can see here, we have a sample of our botanical garden here that we are keeping. And we said the botanical garden is a kind of a place, a reservoir of our genetic resources. Within a place like that, you may bring all the plants within this you have Southern Guinea savanna and put them in the botanical garden, at least for study. So we also have seed banks. If you get to IATA, I think there we have a seed bank. And the first time I went to that place, when I entered the place, I told the man said he will close the door. I said, if I enter this place, I don't think I will survive for five minutes. I don't think I will survive. Because the place was so cold. And they have all the kind of a gene bank for seeds. All the seeds we have, a plant we have in the whole Nigeria, they were deposited here. And that seed can remain there for over 100 years, and they will still remain active after. So it's a method of protecting some of these resources we have, especially those that are trying to go into extinction. You can keep them in form of a seed bank, tissue culture, except botanical garden and herbarium. And herbarium is also here. After our collection from the field, we collect a good sample. And in those days, I used to tell my students, don't collect any plant that is diseased. Collect plants that are vigorous, with good leaves and everything. Pressing. After collection in the morning, we do the pressing. At least we've seen the herbarium press here being shown to us this morning. We press. After pressing for some time, it continues to dry. And I used to tell my students that for you to get all my mark in those days, you know it must be green dry. For those that passed through that program then, after pressing it for maybe like 2 weeks, you will see those plants will still remain or retain their green form. And after retaining it, we do the mounting on herbarium sheets. After mounting, then we prepare the legend containing the data, like a legend containing all information about that plant, the scientific name, the family name, the species name, the characters of the plant, the habitat, the name of collector, the place of collection, time of collection, the accession number. Everything is on the legend. And we place the legend by the right side below on the mounting sheet. After putting them in the mounting sheet, then the next thing is to prepare a folder where we keep them, and then we store them in the main herbarium. We store them in the herbarium. This is for study. Any student that needs any information about the plant, they will come there with their. That is by searching. It's a very tedious process. Very tedious process because you have to bring it out, bring the folders out one after the other and do identification by matching. You open the first folder, check the plant you are holding. Does it match with this? Until you get to that plant. So modern biotechnology has enhanced preservation through techniques such as cryopreservation. That is storing plant tissues, living cell, seeds, or other biological material at ultra low temperature. When low temperature, that is very, very low, as up to minus 196 degrees Celsius. Very low. And it will put all the things within that tissue or plant in a kind of static form. It will remain static, it will be static and remain dormant until so many years when you need it. You now bring it out, warm it, and it will come back to its normal. And it will become normal again, then you can replant it. Okay? So this one help us to keep those things for a long time. If you want to keep any material for a longer time, you can use this cryo preservation. We also have a micropropagation, cloning plants in laboratory to produce large numbers for reforestation or restoration project. So no preservation effort can succeed without education and collaboration. We must train new generation of botanists, ecologists, and conservationists. Communities must be engaged in identifying and protecting local species. Government, universities, and NGOs must collaborate to create effective conservation policies. As scholars and practitioners, we have a duty to combine scientific knowledge with traditional wisdom. Local communities often hold centuries old understanding of plant species and their uses, and such knowledge that must be documented and preserved alongside with a modern science. Okay, I'm concluding now. In conclusion, plant identification and preservation are not just scientific exercise. They are moral responsibilities. We ensure that the green heritage we inherited is not lost, but enriched and passed on. So let us remember that every plant identified and preserved is a step towards a more sustainable and livable planet. Together through research, education, and action, we can safeguard the diversity of life that sustains us all. So may we all continue to nurture the green gift of nature. So I want to thank you all for listening. God bless you.
All right, thank you very much, sir. Please, can we join our hands together for him? Thank you, sir, for that very, very wonderful presentation. Next on the list is a presentation on plant identification, preservation techniques, and also funding opportunities. This will be taken by our guest speaker, Dr. Daniel A. Ziegler. He is a lecturer with the Gombe State University, presently in Germany. So, sir, if you're with us, he will be joining us online. If you're with us, sir, you can go ahead with your presentation. Dr. Ziegler, can you hear me, sir? Okay. All right. While we wait for him, I'm going to call on the next presenter, who is going to be speaking on AI application in plant identification and modeling. That is in person of Dr. Aliyu Salisu, who is from the Department of Computer Science. Please, a round of applause for him as he takes the podium. All right, Dr. Ziegler, can you hear me, sir?
Hello.
Yes, sir. Can you hear me loud and clear, sir?
Yeah, I can hear you.
All right, sir, you have the podium, so you can go ahead. You have just 30 minutes for your presentation. Thank you, sir.
Can you see my screen?
Yes. Very well, sir. Yes, sir. Yes, sir, we can see your screen.
Okay, so I'll start by thanking Professor Ahmadu Bello, Aliu, and the Chairman Organizing Committee, Dr. Dauda, for finding me worthy to present to you at this workshop. Thank you so much for the opportunity you have given me to share my understanding and some literature on plant identification, preservation techniques, and funding opportunities. I would also want to thank the audience for finding time to attend this type of workshops. We hope that by the end of the workshop, you will be well equipped with the aspect of plant identification in particular and botany in general. Some of the topics or some of the ideas that was given to me have overlapped with what Professor Adelowa just presented, especially the aspects of plant identification and preservation techniques. And so I will save some of the stories that I have on this aspect and save some time, so I will be less than 30 minutes. But I will also want to say that I've modified the topic to slightly reflect some of my advocate regarding plant identification, which is that giving voice to silent herbaria in Nigeria. So I wanted to talk about what plant identification is, which is a process of determining the correct name of a plant. So no more stories because my work was made easy. But why do we have to identify plants? It matters because it gives us accurate identification and it gives us like a key to literature of the plants. It supports biodiversity research, it guides conservation planning, and ensures that accurate ecological and botanical studies are done correctly, because if you miss the name of the plant, your downstream analysis or your downstream literature will be wrong. So accurate identification of plants before research, very key to the literature of the plant. And also it helps in tracking invasive species, just like Prof has just highlighted. Therefore, I would like to talk to you on the following outline: innovating traditional applications to plant identification, best practices in specimen identification and preservation, the current state of Nigerian herbaria, my advocate, and then lastly, exploring funding and collaborative opportunities, especially to Nigerian scholars. Classical approaches to plant identification. This aspect Prof has addressed, but the first thing that comes to your mind if you collect plants is to look at the morphology, look at the flower, look at the leaf, look at the fruit, and any other physical observation that you can make on the plants. If you are not able to identify it, the best way I would recommend to call an expert, either the Herbarium curators or people who have worked on a particular group of plants. For example, if I know that Dr. Ahmadu has worked on acacia, and then I collected acacia that I cannot identify, then it is appropriate for me to call him or send him the specimen of acacia and say, I'm sure this acacia, but I don't know the name. So contacting experts is very helpful because they have worked with the plants, they have engaged in several occasions with the plants, so they might know better than any other sources of identification. So that expert identification, I will emphasize that it's very important in plant identification. Taxonomy keys them because they are in black and white, so it has to be for experts, for taxonomists, but very importantly, use of various specimens are depositories of plants and plastic, and so they are very important in plant identification. The use of regional floras or monograph. In Nigeria, and especially in the savannah, one of the key important materials that we have for identifying weeds, especially agricultural weeds, is the long-standing Okobundo and Agueka 1998. There's the second edition or third edition in 2016. It's a very good handbook that one can use for plant identification. Another one is our current book in 2023 that was published, Savannah Herbaceous Plants. It's also a very guide to identifying plants, especially the savanna floras. The second book, the Savannah Herbaceous Plants, is a build up from the Okobundo book, but more expanded in the sense that it did not only consider agricultural weeds, like in the case of Okobundo, but considered all herbaceous plants, either on arable lands or even in the wild. So these are some of the ones that we have, especially in the Savannah area, that we can use for identification. And one important aspect of identification is monographs. So although it's not very popular in Nigeria, monographs are very important because they are very specific to group of plants. So for example, I can decide to prepare a monograph on Asteraceae, and it will go deep into the details of morphological and some anatomical structures of those plants, and they are very key in identification. Today, technology is transforming so many aspects of life, including research and also in plants taxonomy. And so there are modern AI-assisted identification tools that one can use for plant identification. One of them is DNA barcoding, which can be RPCL or MATK or ITS. And so one can use this. Another platform that you can use for plant identification are the digital ones like iNaturalist, which tomorrow we will talk deeper into. So I don't want to preempt our practicals tomorrow, but iNaturalist is one of the important digital platforms that one can use in plant identification. Other apps that one you can use for plant identification is PlantNet.
So Ziegler, sir, can you hear me? Yes, sir. Your slides are not changing.
They're not changing.
Yes, sir.
Okay.
Yes, sir. Can you please change them to presentation mode?
Are you seeing my slide?
Yes, sir. We can see it now.
You can see the title of the talk?
Yes, sir.
Okay. Now I'm changing it to presentation. Is it changing?
Yes. Change, let's see, sir. All right, it's fine, sir.
Is it changing now?
Yes, sir. You can go ahead.
Is it changing? Yes. I want us to move to the aspect that we have missed. Is it changing the outline?
Yes, sir.
What about this?
Yes, sir. It's changing.
Okay. I think this is where I am, right?
Yes, sir.
the use of digital platforms. Okay, so like I said, I will concentrate on the funding aspect because the identification aspect has been addressed by Professor Adeloma. I want to emphasize that you cannot tell a funder that you want to do identification, so give me money for the identification. It will not be possible because you have to convince your funders about what you want to use the money for you to get funding, especially with national organizations. Money is very difficult now, so you have to take your time to convince whoever will give you money to fund your project. Plant taxonomy in general and plant identification, people consider it as very dry, or that it's very difficult to convince people on giving you money to fund your project. Well, I differ with this because this is one of the reasons for this workshop, to be able to tell us that there are some opportunities out there in plant taxonomy that will give you money to fund your research. But then what you have at hand will determine whether the funder is willing to give you money or not. So herbarium collection is one of your currency, is one of the things that will give you money. So as the process, the first thing you do is to go to the field by yourself and collect a different collection process. The second one is you can decide to put these plants, once you have them imaged, to put them on digital platforms, like the ones we showed, either on iNatural, on PlantNet, on GPIF, or on GPIF image IP. Or you can take the material, the herbarium, and prepare them, just like we have mentioned, using the comparative approach of comparing your sample, your specimens with ones in the herbarium, and then you deposit the material in the herbarium. Over time, this collection becomes your currency. Why does herbarium matter? Or why do herbarium matter in research today? Herbaria are libraries of plants that they are essential for biomagnetic science. And there's a quote or adage by one of the scientists that is a nation without an active herbarium cannot know its flora. This is because herbarium specimens have moved away from the traditional taxonomic work to more advanced approach. On this, you can see about 25 areas that use herbarium specimens for their research. So if you want to do research in the lab, in the herbarium, it's very possible. But we have to have a wealth of collections in those herbarium. Herbarium is no longer just for taxonomy, but it's for biodiversity science. Any area of science. The one I know is that we use herbarium for palynology, we use herbarium specimens for anatomy, we use herbarium specimens for basic descriptions and so on. But the world has moved on into other aspects of science using herbarium materials. So the evolution in the use of herbaria, because of high number of digitization, because of large number of teams coming together to work as a team. So for example, different institutions can collaborate together to work on herbarium specimens across the globe. And new domains of experts. So people are becoming very restricted to a group of plants. So let's for instance, I'm working on Herbaceae, someone is working on Asteraceae, someone is working on Poaceae. The new domain of experts in different areas of plants is transforming how herbarium specimens are used. And so because of this team, you can see that the globe is becoming a meta-herbarium. So you no longer do your work in your herbarium and then you don't connect to other herbaria. So the world is becoming like a global village, and the herbaria are becoming like meta-herbarium, becoming one. And so there's need for this collaboration, and so there's increase in the number of specimens that are used for scientific purposes across the globe. The next image that you see by your right is the number of specimens in millions, and this has grown across the globe. From 2010, when GBIF was launched, more materials are deposited, more specimens are on GBIF, and more information is out there. So this is connecting the globe into one herbarium that today you can use herbarium for almost every aspect of biodiversity side. I just want to connect with the audience. Are you following the talk? Yes. Okay, thank you. So the use of herbarium specimen is expanding the scope of biodiversity science. And I would like to draw our attention to the publications that are coming out of herbarium specimens or into herbaria. From literature, plants that publications that are emerging from use of herbaria are growing every year. You can see the exponential of publications that are coming out of herbarium. The question I want to ask the audience is, you can see that publications from herbarium specimens are growing across the globe. How many publications are coming out of our own herbarium? So for example, ABU Herbarium is one of the biggest herbaria in Nigeria, or probably in northern Nigeria. How many publications are coming out of that room? If you want money to come into the herbarium at ABU, because you have to convince your funder that you are using what you have, and then use that as you leverage on those publications that you have used, produced from that room, to be able to tell your funder and you want more money to be able to produce more literature or contribute to knowledge. So we begin to think towards the aspect of the use of herbarium to produce knowledge, and then ask for funding from whoever would potentially fund them. I want to draw our attention again to where Nigerian herbaria are in global discourse. If you look at figure A, it shows country-level representations of the number of specimens deposited in each herbaria across the globe. And you can see that most of the countries with 30 million specimens are in North America or in Europe. In Africa, the bluish part, which is always in Africa, which means that most African countries have specimens in their herbaria that are only between 5,000 to 200,000. And in Nigeria, you can see Nigeria is at the dark spots, which has 5,000. The maximum specimen in Nigeria is 200, according to this record. It is only South Africa and part of Kenya that are in yellow, which means they have 1 million specimens. So Africa is lagging in terms of economic discourse. And then I streamed down to Africa and looked at what are the 20 largest herbaria in Africa and how many specimens do they have. Clearly, the first 10 herbaria in Africa are in South Africa, East Africa, and some part of Madagascar. Nigerian herbarium, that is a forest herbarium Ibadan, which holds 105,000 specimens, is number 15 in Africa. That means that Nigeria were not doing well in terms of collections, in terms of the size of our herbarium. And that follows by Ghana in West Africa. Of course, we are still the giant of West Africa. But then it comes to the African discourse on herbarium, we are not actually doing well. So it's a sign that we need to do well more in terms of specimen collection. Where is ABU or any university in the north in Nigeria? We are not anywhere close to number 50 in African discourse, which to me, I find this as a challenge, and therefore use this medium to call on herbarium collectors, herbarium curators, to increase more effort in terms of collections. If we want money, if you want funding, if you want people to come and give us money for research. In one of our recent papers, the Silent Herbaria, we looked at how many herbarium are in Nigeria. There are about 5 herbaria in Nigeria. Of these 55, only 17 are registered in the International Herbarium. And you can see that all herbaria in Nigeria are classified according to international standard as very small, because all of us have less than 10,000 specimens in our herbarium. And so it's difficult for us to attract funding. Again, of those that are registered index herbarium, only 5% were able to integrate their collections into global repositories like GBIF or JSTOR global plans. And so it's a huge.
All right, while we wait for the network connection, I would like to introduce some guests that just walked while the program was on course. We have Professor W.S. Jaffet, that is the Director of Species. Please, a round of applause for him. We also have Professor M.S. Mohammed, that is the Head of Department Plant Science. Sir, can you please come up stage? Please, a round of applause for him. Have a seat. Thank you.
Okay. Can continue.
But can you see my slide?
Yes, sir.
Okay. Can I continue from here?
Yes, sir. Go ahead.
So I was talking about.
You have 5 minutes, sir.
And their specimens. And I said, the largest herbarium in Nigeria is that of Flynn, which has 100 and is number 15 in Africa. The rest of the herbaria are in South Africa, East Africa, and Madagascar. So I was saying that we need to put more effort in terms of collections if we need international funders to support us. I also talked about the fact that we have 55 herbaria in Nigeria, and 73% are not registered. Nobody knows about them. Nobody knows that they are existing. And our studies found out that 100% of all Nigerian herbaria are small herbaria because we have less than 10,000 specimens in each herbarium. And only of those that are registered on Index Herbarium, only 5% have integrated their data into global repositories or just called Global Planet. And so it makes our work to be limited in terms of visibility. And so if you want to confirm herbarium in Nigeria, it's difficult because it's visible. However, we are doing well because 88% of our herbaria are used for teaching, and it means that we are contributing to knowledge in one way or the other. And we also looked at the origin of species that are in this herbaria, and we found out that about 10% of our collections are native to our localities. That means that some of them are not found elsewhere, only in our local region, and that means that our herbaria are contributing to regional geography or regional biodiversity science. So it's a matter of putting this specimens visible. We can make contribution to science or to knowledge. So our herbaria needs updates. We need to upgrade them because they are facing a lot of challenges. And one of these challenges is funding, and that poor funding leads to poor facilities and pest management. And I shared with you one of the herbaria that we visited in northern Nigeria. I don't want to say the name, but this is how it looks. And imagine the way it looks is not actually good. So there's these poor facilities at levels Nigerian herbaria. And then staff training. Staff are not trained in botany or herbarium curation, and they are being sent to this herbaria. For example, I mentioned that we visited a herbarium that the curator is a microbiologist. So I asked him, how did you come here? Because you're a microbiologist. Are you studying the fungi on the plant specimens or microbes? You know, if it's possible. But that goes to the extent to tell you that Nigerian herbaria are not getting correct staffing that it requires. And so this together makes our collections be invisible in terms of global network. No digitization facilities is a problem in Nigeria. Ideally, this is how it should look, where a conveyor belt to digitize the specimen if you have 1 minute, within some days, you'll be able to digitize your herbarium. What we have in Nigeria is something like this, lack of collection culture. Most of the collections that we have are student collections. And student collections, you can see, is either on a wrong paper, you see the specimen extending beyond the recommended paper size, or there is no collection notes on the specimens. You can see this here, it's just said Albizia lebe and no any other collection information. Or the collections are just clumsy in terms of mounting it. So students will just say, tomorrow is submission date. They will just collect the plants from the bush and iron them and bring them to. So our herbarium over-reliant on student assignments. And so no legacy in terms of collections. And so our herbarium specimens are very not in the shape that you can be proud of sharing it on global networks. In one of our studies, most of the specimens that are deposited in this herbarium, because they are student assignment, they are plants that are being cultivated, especially the cowpea, the Fabaceae species, granules, beans, bambara granules, are some of the specimens that are always represented in most of our herbaria. Or the poaceae, the grasses, guinea corn, millet, maize, sugar. The common grasses in this area that are around are the common specimens that are found in this area. So if you look at the kind of collections in the area in Nigeria, you will find out that because they are student-based, most of the specimens are cultivated because they are collected around households or around house. So this also contributes to some of the challenges that we have in Nigerian herbarium. Although we have some challenges, there are some signs of progress. One of the signs of progress is an exponential increase in our collections. You can see over time from past independence, there was increase in the annual number of collections in Nigerian herbaria, which is also more skyrocketing post 2020. So it's a sign of progress and that we are also making some effort. So there is hope for us. Also, there's emerging young taxonomists or herbarium curators that are making effort in the collection. So, especially in the aspect of collaborative digitization, the Savannah project, for example, have added to our collection. And there are other partnerships that we get from other funding organizations is making our herbaria to be active. So if you compare the growth of herbaria in Nigeria with that of the global trend in golden color, for example, is growing slowly. And the one in red, which is Nigeria, has grown very deep because of the effort that these young taxonomists are putting in place. And so there is this hope that we have that the growth of Nigerian herbarium that is exceeding the global trend. So it's a hope that we have in Nigeria that will propel us to the next level. Of collections that we have in Nigeria, like I said, is the source to our funding. But some of the information are missing in Nigeria. I gave this slide to show you how collection gaps are found in Nigerian collections. So for example, this is the global distribution of hibiscus versifolius that were collected in Africa, which is native home to hibiscus. And if you look at the collection, we have some records of hibiscus versifolium, Cameroon, our neighboring country, Chad, Niger, West African countries. But clearly you can see Nigeria is missing. One then asks, is it that the specimen or the species is missing in Nigeria, or there is no collection in Nigeria? So in one of the studies we actually conducted that, we found out that we actually have the specimens deposited in our herbarium, but the global network did not know about it. So this and many more are some of the challenges and strengths that we have. As part of the funding opportunities, I've listed some of the funding organizations. The first one is to look inward, in-house. What are the national opportunities that we have? Most of our herbaria are in the universities. So the first thing is to be able to explore national fundings for herbarium. So national fund would be IBR, which is institutional-based research grant from TED Fund, which I think is about 5 million naira. And I think it's sufficient to start the project. It will not complete, but it will start the project. You can buy at least one camera and one imaging station, and then you explore other bigger funding like NRF, National Research Fund. So the first one is the national one, the one we have locally at home. So please explore that. Second one is the International Association of Plants Taxonomy. It's a small grant, $2,000. I don't know if they have increased it now, but the last time I accessed it, it was $2,000. But it's enough to at least start the basic digitization process or the basic collection effort that will increase the number of collections that you have in your herbarium. And again, there's another funding from AIPT, which is about $10,000 for small research grant for smaller herbaria. So if your herbarium is less than, if your herbarium has less than 5,000 or 10,000 collections, you can access that grant. So $10,000 is enough to get more materials into your herbarium. So it's an opportunity again to access that. So apart from $1,000, IAPT can provide. There's another one that is 10,000 that one can access once you can put your ideas together. GBIF, BID, BFA, BIFA is also an opportunity. Last month there was an advert, I think it has closed early this month, and it gives about 30,000 euros to institutions. So it's for ABU, for example, put the biodiversity resources together and provide a strong reason for digitizing its resources, and they get funding. So GBIF is one of the funding organizations that can help digitize your herbarium. Pod Foundation is very specific to species, but you can use herbarium material to prove your point that species are actually that they need intervention. Mohamed bin Zayed Fund is also very Nigerians to preserve their biodiversity, and herbarium solution is one of the sources of funding for them. So to write a proposal, the first thing is you need to prove that you have collaborators, either within the country or abroad. So if you just say you are on your own, you want to do it alone, then the next idea is why you alone. So the world is becoming more collaborative in approach. So the national and national cooperation can make a proposal. Your budget is very important. If your budget is realistic and has
now, why plant identification modeling matters? For centuries, botanists have depended on visual observation and taxonomic keys
to identify plants. This method, though rigorous, is time-consuming and limited by human expertise. Herbaria houses priceless specimens, but many remain physical, underused, and vulnerable to decay. The question is, how can we digitize this rich heritage, make it searchable, and link it to living ecosystems? The answer lies with us. Not only that, preservation. Digitize historical data and subtle morphological differences far faster than manual methods. Digitize Herbaria protects historical data from physical deterioration and make it what? Searchable. Thirdly, we have connectivity, linking specimen to GIS and peace distribution and ecological change over time. With this, you may want to ask, what exactly is this? Many of us may think when we hear AI, it represents, it represents here. You are not far from the truth. This is almost correct. But what drives this robot is the AI software that drives the robot. Take, for example, take a look at these AI-driven robots. Play. It's not playing. Click on that button. No. Yes. It's not playing. Okay. Anyway, so click on the slide, the main image, then click on the. Yes. Okay. It's unfortunate that it's not playing, but these are AI-driven robots showing their skills in football, able to identify where the ball, the position of the ball, and that is able to see objects and identify objects. Not only that, they are also able to maintain their focus to see which direction they need to play this ball to. The next slide also shows, next slide, the world record attempt for the farthest basketball shot by a robot. Unfortunately, this is not playing, but we have robots being trained to be able to play basketball in such a way that as soon as the precision is high and they're able to throw this ball and it gets into the net. And we have a world record of the longest or the farthest. You want to try that again? Okay. The next one, just click on the next one. Yes, click on the next video, just play that quickly. This illustrates the farthest distance ever. And the next slide also, this is robot in action, AI-driven robot in a boxing match. Quickly. Now you can see he knows his opponent and knows what is happening. And even when he's down, he responds to the counting from the coach. So this is to tell you how far AI has gone. Next slide. Now, this is also AI. AI we can use together, we can use these robots as, you can play that quickly. No, go back. Try to play that quickly, go back. As a companion to help us in our harvest. So we have robots harvesting. Now, what drives this? Next slide, please. What drives this is not just the robots, but the software in them. We have come to the stage where robots are lifting heavy objects in construction sites, performing repetitive tasks, taking roles that poses threats to human safety, like mining. So take note, the next time you see a robot, just note that it may be smarter than you. 2, they serve as personal assistants nowadays. They manage your schedules, they manage complaints requests as customer service agents. So note that the next time your device seems to read your mind, don't be surprised. Next, AI serves as an astronaut. The role of AI in space has expanded. They are able to analyze their environment, adjust their course, and even conduct experiments without human intervention. So the next time you look at the stars, remember that AI might just be looking back at you. On the road, we have AI. Ever dreamed of a car that drives itself? Dream no more, as self-driving cars have shifted already from science fiction to reality. And we also have, finally, AI as our environmental guardian. Picture AI system sifting through mountains of climate data, identifying patterns and predicting future trends, giving us a chance to intervene before it is too late. Coming back to our discussion today, if AI can do all those, do you agree with me, even before going further, that AI also have a role to play as a botanist? Yes. AI refers to computer systems that learn and make decisions from data. In plant science, that means teaching machines to recognize leaf shapes, predict flowering, right? Predict flowering and simulate emphasis, just as botanists would, but faster and at a larger scale. AI has some subfields of learning. We have machine learning as a subfield or pattern discovery. We have deep learning, especially the convolutional neural network, for visual tasks to be able to understand images. We have computer vision to enable machines to see plants, but with greater precision. This is AI. How does AI do all this? This is exactly how we do it in AI. You have a herbarium, a collection of plants, images. Look at this diagram carefully. This is the whole story. For any AI software or tool you may be using, this is where the journey started from. We have a collection of digital representation of plants as images. We annotate them, that is, we label them properly. It undergoes some sort of training. This training is managed by specialized algorithms that understand these images. And from these images, it will be able to predict a particular type of plant or leaf. Given any part of a plant, to be able to identify that particular plant. This is the whole story behind identification of plant. Next slide. Now, AI, we have seen has its place in plant identification. One of the earliest and most impactful uses of AI in botany is plant identification. By training deep learning models on thousands of labeled plant images, we can now automatically identify species with remarkable accuracy, even from partial or damaged leaves. AI has a role to play. A good example of an AI tool is the PlantNet, an AI-driven platform that allows anyone to photograph a plant and get instant identification using AI models trained on millions of images. Similarly, another example is the leaf snap. The leaf snap is also another example of an AI-driven software that can be used to identify plants, making you not rely on the manual process. Another example, again, is the flora incognita, The Flora Incognita is another software that leverages the convolutional neural network to classify plants in real time. These tools are now empowering researchers, farmers, and students, hence reducing dependence on limited taxonomic experts. Beyond convenience, they contribute to biodiversity monitoring and early detection of invasive species. Today, we can even embed AI in mobile phones and handheld devices. Field researchers in remote areas can identify plants offline. No internet required, thanks to edge AI computing. When linked with geographical information systems, these applications can map plant species distribution across the entire ecosystem. That is AI in plant identification. How about AI in modeling? From growth to forecast. While identification is powerful, modeling is transformative. AI can simulate how plant grows, how they respond to stress, and how climate change can affect them. All this you can do with AI. For instance, the long short-term memory algorithm can predict flowering times, fruiting, and yields using temperature, rainfall, and soil data. Other models, other algorithms, like the random forest, the gradient boosting algorithms, all help in forecasting crop productivity or detecting diseases from satellite data. AI can even model the spread of plant pathogens, predicting when an outbreak might occur and how it could affect yields. These models are invaluable in agriculture, forestry, and conservation, giving us foresight we never had before. Now, AI and Herbaria digitization, these are two things we need to take seriously. Digitized Herbaria are the new gold mine of plant data. Let's take note of this. Digitized Herbaria are the new gold mines of plant Herbaria. Herbarium sheets and fed them into AI systems. We create training data that can identify species, recognize structures, and extract metadata from optical character recognition. This fusion of historical data and modern AI brings the past and the present together, helping us track changes in plant morphology, migration, and even climate adaptation over centuries. Next slide. Case studies and success stories. Let's highlight a few word examples. An AI tool known as the Plant Village Nuru, developed by Penn State University, uses AI to detect crop diseases from phone images already deployed across African countries now, as I speak. The other software, Flora Incognita, identifies more than 4,800 species in Europe using AI. In the Amazon, deep learning models have been used to map real species from drone images. These breakthroughs shows that AI is not just a lab tool, it is a field companion, a conservation ally, and an educational partner. All these successes are not without challenges. We have challenges, especially in this part of the world. Of course, challenges exist. Our data are often biased towards certain region and species. And this is why when you use some of these tools to identify our local plants, you see that they fail because they were not trained with those particular data sets. Our collection is localized. We need our own AI-driven software to be able to identify our local plants. If, for example, I have an AI system that to detect skin cancer, this AI system is developed by professionals out there that only recognize white skin. You will agree with me that such software will definitely fail when it comes to the black man's skin, right? Same applies to species that are peculiar to us in this part of the world. Not only that, the quality of our data sets may affect how well this AI system works. Variations in lighting, background, and camera quality can mislead AI models. So if you are using an AI software to detect plants and your camera is not good enough, so do not expect a very good result, right? Across access to computational results also, and expert-labeled data is still uneven across the globe, especially in the global south, right? But each of these challenges is also an invitation and an opportunity for collaboration. By developing localized data sets and fostering partnership between botanists and AI engineers, we can build models that truly understand our flora. Future direction. Next slide. The future is exciting. We are moving towards multimodal AI, combining images, genomic data, and text. With federated learning, global institutions can train AI models collaboratively without sharing sensitive data. And with generative AI now in place, we may soon simulate plant growth under hypothetical climate conditions, a tool for both research and policy. In conclusion, artificial intelligence offers us more than tools. It offers us a new lens, a lens through which we can see the unseen patterns of nature, protect endangered species, and optimize our agricultural systems. But the success of AI in plant science depends on one thing: collaboration. Botanists, AI engineers, scientists, policymakers, and institutions must work hand in hand. Let us digitize our herbaria, share our data sets, and build intelligent systems that preserve our planet's botanical diversity for generations to come. Note this: the plants of tomorrow will not just be observed, they will be understood, modeled, and protected through the power of artificial intelligence. What we are trying to do, in essence, is that for botanists here, we are not trying to steal your job. We are trying to say that use AI to make your job easy. And for those that are non-botanists or non-plant scientists, what we are saying is that the next time you have the choice to make between two surgeons, where one is an AI and the other is a human, feel free to take the AI surgeon. Thank you.
Thank you very much for a wonderful presentation. Please, let's appreciate him again. Let's appreciate him. All right. Now we're going to take question and answers. Questions from the first presentation made by Professor Adelawan, second presentation by Dr. Daniel Ziegler, and the last one by Dr. Aliu Salisu. If you have any question, please. Question? All right. In the absence of any, we're going to take the vote of thanks by the chairperson, local organizing person of Dr. Suleiman Dauda. Please come up stage.
Distinguished Vice Chancellor, ably represented by the DVC admin, academic rather, Professor Raymond Bago, the head of department, our keynote speakers, Professor Emi Adelawa, Dr. Daniel Ziegler, Dr. Aliu Salisu, facilitators, invited guests, participants, ladies and gentlemen. Good afternoon. It is my singular honor and pleasant duty to deliver this vote of thanks on behalf of the organizing committee for the workshop on plant identification, herbarium digitization, and the application of artificial intelligence in plant characterization, organized by the Department of Botany, Faculty of Life Sciences, Ahmadu Bello University, Zaria. First and foremost, we are deeply grateful to the Vice Chancellor for his